for those of you who haven't heard of Primrose Bio before, we were formed just a few years ago in 2023. Oh, sorry. Adjusting for short people. All right. From the merger of the Pfenex team with Primordial Genetics, which is a synthetic biology company. Pfenex was a group that was working on protein expression in P. fluorescens. And we actually have a really long history with the Pfenex platform. We're located in San Diego, and we focus on strain engineering using Pseudomonas fluorescens and have capabilities to support strain engineering all the way through process development, analytical development, and getting to enabled GMP manufacturing. So why Pseudomonas fluorescens? Well, really, we isolated this organism because it was great at making proteins. So this is a very purpose-built platform. It's designed for expression. We didn't take something from the lab and then try and shoehorn it into making proteins. So once we identified this organism, we built a toolbox around that. So applying combinatorial strain engineering, using a multi-omics approach, we've been very focused on how to get this organism, which is a great protein expression organism, to make soluble, active, properly folded protein. And we'll go through some of the features of this toolbox and the organism and why it is, or we feel it's so great. So a little bit about the platform and the toolbox as we've built it. We have a number of off-the-shelf plasmids with typical features, so different promoters, different ribosome binding sites, etc. So we can tune transcription, translation. Another really important feature is our large collection of signal peptides. Pseudomonas fluorescens is a gram-negative organism, so it does form disulfide bonds in the periplasmic space. So we need to get protein out there to make sure that it's properly folded in those cases. And we combine that with an array of different host strains. So these are engineered to help proteins to fold properly, really focused on quality. So we've got a number of folding helper overexpression strains, protease knockouts, things that can reduce nibbling and chewing of your protein of interest. And then more recently, we've been focused on some more advanced tools for non-canonical amino acid incorporation, other tools for improved folding, and then streamlined recovery. And it's the non-canonical and streamlined recovery aspects that I'm going to talk about here today. And just to give you a quick overview of how we do most of our screening campaigns, Pseudomonas is very well adapted to growing at very high cell densities, even in a 96-well format. So we do all of our screening in 96-well half-mil cultures. We can do this in different modes. This is describing our FAST workflow, where we use a pool cloning approach, taking maybe one optimized gene, encoding the protein of interest, and then combine that with a number of our expression vectors in a pooled cloning, plate those out. Under selection, we do use complementation for our selection. So everything is antibiotic free, and our media is totally animal origin free. It's just a mineral salts media so that we can do this selection by complementation. So after we grow these up, induce them, we can then lyse, rank those individual cultures by titer using either CGE or ELISA, and then identify our hits, enrich those, and move on to additional analyses. So all of this can be done from gene through this initial enrichment in about 10 days. So one of the things that we felt would, of course, very much streamline this workflow is if we can move the protein from the periplasmic space out into the culture media. And you'll see in later slides me referring to this as CFB or cell-free broth. That's just kind of our internal lingo for describing protein that we get outside the cell. So why extracellular? Obviously, it makes it really easy to recover, but we want to make sure that we are focused on high quality protein. So for us, it was really important to do a paraplasm first strategy where we first get the protein into the paraplasmic space, allow it to fold, allow the disulfides to form properly. Sometimes, as you know, this can take a little bit of time, especially for some of the fabs that are out on the market. These are slow folding, slow to assemble, and we want to make sure that that is all happening properly before we move it outside the cell. So in my first case study, I want to talk a little bit more about data with a number of different proteins that we've screened using our different extracellular localization host strains. We wanted to apply this first in a 96-well screening format to see how can this help in the discovery and development workflows. It was important for us to make sure that this was flexible. Proteins come in all shapes and sizes. So we looked at proteins of different types and ranging from as small as 15 kilodaltons all the way up to over 120 kilodalton complexes. So how we did this, first we looked at different targets that could influence movement of protein into the extracellular space or cell-free broth. And then we wanted to ensure that these proteins that we were moving were active. So we're going to look at some activity data and that they are of good quality. So we want to make sure that they're intact, that those paraplasmic secretion leaders are cleaved properly, and that all of this works well in that 96-well format. So in our first example here, we're looking at a multimeric enzyme. Shown on the left are the titer results that we achieved. So here we're looking at 3 different host strains. So these are differently engineered, and they're just labeled here EXC 1, 2, and 3. You can see from the CGE results, the cell pellet side, this is just lysing cells that we've spun down. This is protein that's localized into the paraplasmic space. We're expressing the protein well in each one of these hosts. Wild type is just our standard Pseudomonas fluorescens. And what we can see is that how we engineer the cell for extracellular localization impacts how much protein is getting out of the cell. In this case, EXC001 is giving us the highest amount by CGE titer. When we take that culture supernatant or cell-free broth and then evaluate the enzyme activity, we can see that the amount of protein we have around correlates to the amount of activity. So we're getting the most protein out with EXC001. That's the one that's giving us highest titer. It's important to note that the tetramer formation is really important and required for activity. So we verified that we're getting something out that's over 120 kilodaltons. So very impressive for microbial, for getting a protein that large out. When we did intact mass, we saw that the secretion leaders are cleaved as expected. And we can envision how we can use this in a workflow for enzyme engineering, which is another thing that our team works on the synthetic biology side. So that if you normalize this by mass and load the same amount of protein into your enzyme activity, you can look at differences in specific activity in an enzyme engineering campaign. Now, in the next example here with high throughput data for our extracellular strains, we're looking at a couple of different antibody derivatives. Here just labeled antibody derivative 1, 2, and a binding peptide that we've fused to one of our collection of fusion partners that we use for peptide expression or toxic protein expression. So again, looking at the CGE data, we can see that different extracellular host engineering strategies work in different ways for different targets. If we look at the cell pellets, we're expressing the protein in most of these strains. So we're having some impact on protein expression as well. For the two antibody derivatives at the top here, you can see that extracellular hosts 2 and 3 seem to be working better for these two types of proteins. For our binding peptide fusion, it looked like actually extracellular host number 1 is giving us high extracellular titer, so in the cell-free broth here, compared to the protein that's retained in the cells itself. So again, seeing here that these engineered hosts can enable extracellular localization, and that our high throughput growth and expression system is working with these hosts. Now, next step, of course, we want to ensure that the proteins are actually active and that the quality is just as good as what we're seeing in the paraplasmic space. So this is just an expanded SDS-CGE analysis set here. Again, the same CFB and cell pellet titers that we were seeing on the previous slide. But now if we take the material and enrich it from the cell-free broth and also from the cell pellet, then we can evaluate the binding activity. And you can see here in this curve on the right side that the relative potency of each one of these fusion peptides is pretty much the same. So the quality of protein that we're expressing is very good. Again, proving out the system that if we can get protein out, we're getting very nice, high quality active protein out of the paraplasmic space. The other nice thing is once we enrich this protein for these fusion peptides, we can then readily cleave the peptide off of our fusion partner and then evaluate it in further assays downstream. So hopefully what everybody's been able to see is that with this extracellular localization, engineered host strains, we can really streamline the discovery workflow. So we can use these strains earlier in the pipeline for hit characterization, lead ID optimization, and really streamline that expression and generate a good amount of material so that additional assays can be performed earlier in those steps. So what we're seeing really with the extracellular localization is about 50 to 80% of the proteins getting moved out into the cell-free broth or the culture supernatant. The other thing that we've observed in some other experiments not shown here is that we can tune our expression strategies with these different host strains to be able to maximize the amount of protein we're getting into the extracellular medium. So now switching gears a little bit to some of our work with non-canonical amino acids. This is something that we've also integrated into our high throughput screening as well as into later development activities where we've engineered Pseudomonas fluorescens with various tools for incorporating non-canonicals. So we have different synthetases and tRNAs that could be used to specifically insert non-canonical amino acids into amber positions within the coding sequence. I'll show you in a couple of slides that we're able to scale these up. So when we achieve very high titers of protein in fermentation with different molecules, we can maintain that titer when we add the non-canonicals, and these proteins are functional. So you can see we use a very typical workflow for evaluating and screening different non-canonical insertion sites. So we use molecular modeling or HDX mass spec to evaluate different sites on a protein that could be good for inserting the non-canonical, then insert amber codons at those specific sites in the coding sequence, express those in engineered Pseudomonas fluorescens strains that have the synthetase and tRNA pair that we want for inserting that particular amino acid, and then follow that up with confirming that we can conjugate those. So in this first case, we're looking at VHH ADC candidate that we have been developing. We evaluated a couple of different sites on the VHH, so position 84, 98, and 99 for inserting PAZF so that we could do click chemistry and attach a drug target. When we looked at expression in 96 well, we could see that these 3 sites all were able to allow insertion of PAZF. We had also evaluated position 64 and found that that one did not work very well. So always good to be screening a number of different sites. The nice thing is at the 0.5 mil 96 well scale, we were able to get soluble titers of VHH with the PAZF over a mg per mil. When we look at these by intact mass, we confirmed that we have 100% incorporation of PAZF and no native amino acid, so no tyrosine or threonine was observed. So we picked the position 84 for our further development. When we scale this up into high cell density fermentation, on the left-hand side, this is our canonical or wild type VHH. We got very nice high titers of this VHH in our initial 2-liter experiments, so making about 15 grams per liter. And when we look at how do we want to feed PAZF and fit that into our typical fermentation process, we found, not unexpectedly, if you add a lot of non-canonical amino acid, sometimes that can be detrimental to your culture. So high levels of PAZF are not good for protein expression. What we did find is that tuning the amount of PAZF we add and having a continuous feed of PAZF along with our continuous carbon feed, we were able to really quickly tune and be able to show that we can get about equivalent levels of non-canonical amino acid incorporated VHH expression, so about 15 grams per liter, which was really great to see. So no titer hit. We then went forward, purified the material. We showed that, again, we see 100% incorporation by mass spec. When we look at the conjugation of various different sizes of molecules we can attach, we saw really no issues with attaching either large or small molecules to the VHH. We were able to maintain the VHH activity, which is really important. It's great to be able to put PAZF into the molecule, but it's also very important that once you stick something on that, it's still going to be active for your target. So here, showing that with just using a fluor, we're able to see similar potency between the wild type or canonical VHH and the one that has the Alexa fluor conjugated to it. So our next steps with this program, of course, are to develop this molecule conjugated to MMAE or other drug target, then do our in vitro and in vivo efficacy studies. We wanted to make sure that this was, applicable to lots of different proteins. So not just GFP, not just VHHs. Here, another important molecule for Primrose is a vaccine carrier protein called CRIM197. And what we wanted to evaluate is whether, incorporating PAZF and doing that conjugation would enable us to develop more precise controlled conjugation for CRIM. So we evaluated the CRIM197 crystal structure, as well as, again, doing some HDX-MS analysis to see where are there solvent exposed areas where it would be good to insert PAZF for conjugation. CRIM197 is used as a carrier protein for like different carbohydrates and things to really boost the immune response to those carbohydrates or polysaccharides. And so we wanted to avoid any published T cell epitopes in CRIM. So that was another thing that we were looking at. CRIM forms a dimer, also wanted to avoid the dimer interface because we didn't want to disrupt that. So we identified 11 sites for conjugation, did the cloning, inserted amber codons there, and ran this in our PAZF Incorporation expression hosts. And on the left-hand side here, you can see at the 2-liter scale, so this is in high cell density fermentation, we're evaluating what does our titer look like compared to our canonical or wild type CRIM strain, which here is shown as control. And you can see that in 10 out of the 11 places where we inserted PAZF, we got really nice expression, at least or in some cases greater than what we see typically with CRIM 197 expression in Pseudomonas fluorescens. Of course, we wanted to make sure that the protein looked as we expected. So if we purify these different variants with PAZF incorporated at different sites, we can see that intact mass shows us that we've got a single site of incorporation, and PAZF is inserted where we expect it to be. If we then do the SPAC reaction, we can show that it's functional. We can click different molecules to CRIM 197. So of course, our next steps now are to evaluate immunogenicity of the carrier protein with these different insertion sites to see whether these would be good candidates for further development. So hopefully you guys have been able to see today that Pseudomonas fluorescens is a nice strain for protein expression for discovery applications. We can evaluate a number of different variants in 96-well format, get very high titers of functional protein really quickly. A little bit more about P. fluorescens. I mentioned earlier, it is a gram-negative organism. Another really great feature of Pseudomonas is that it's an obligate aerobe. That really helps us to achieve these high cell densities without a lot of the negative effects of going into anaerobic fermentative growth, either at large scale or small scale. It's got a proven safety profile. The other advantage as we go into scale, and I haven't talked too much about our scale-up capabilities, but we do have a number of products that are on the market made using Pseudomonas fluorescens. And the other great advantage is that it gives a really low cost of goods with high titers, a really inexpensive mineral salts media, all animal origin free, antibiotic free. As I mentioned, we do have 6 products that are on the market, FDA, EMA, WHO approved, using Pseudomonas fluorescens as the expression platform. We've got a team that's very capable of scale up process development. We've done well over 25 different tech transfers around the world and worked with a really broad array of different proteins. So it's a very flexible platform. And if you'd like to learn more about it, please stop by our booth, or Russ Coleman also has a poster today and tomorrow where you can learn more about non-canonical amino acid incorporation using Pseudomonas fluorescens. So thanks, and I'll take any questions.
Questions for Diane? I'm going to start with one.
Okay.
I heard a new term, so you're going to have to educate me. Fusion peptides. What are they? And then we're going to go next thing. Why difficult to express?
Yes. So fusion peptides. One of the things I didn't talk too much about in our tools is we have a number of different fusion partners that we use for boosting expression of peptides or toxic proteins. So for peptides, these are in large volumes, get very expensive to make synthetically. And so one of the ways to reduce some of the costs and the waste associated with synthetic or chemical synthesis of peptides is to look at recombinant expression. And so that's another thing that we're focused on as a tool with our system to be able to make these newly evolved peptide drugs that are becoming more and more popular, either as binding moieties or other drugs for metabolic indications and things like that.
Okay, thank you. And again, greener, cleaner, larger quantity. Okay.
Exactly.
Any other questions? Well,
Okay.
we're going to go, your booth is number 624.
624.
And 624?
624, yes.
Yep. And you will be able to see Diane and ask further questions this evening during the reception. So let's give her a thank you one last time for hosting this lunch and technology workshop.